Hello! Today we're going to talk about the cell division or the cell cycle. Cell division is the process that produces two cells from one. And the life cycle of a cell begins when that one cell forms. And it kind of lasts until that cell will reproduce and divide into their two new offspring cells. Before we can talk about the actual process, we need to understand that the DNA can exist in three different forms as genetic material. We talked about chromatin when we talked about the cell parts and the cell structure. Chromatin looked like the spaghetti, so it was in the nucleus, that was the bowl of the spaghetti. So it's very loose, it's very, kind of hanging out in the nucleus. Well, the chromatin will start to condense and form a chromatid, and that is either a sideways V or a V upright. Okay, we also have a chromosome, and a chromosome is two chromatids put together, or an X, that's held together by a centromere, and that's what the centromere is, this little dot that connects the two chromatids. Okay, so cell division is going to produce two cells from one, and it's broken down into two main phases. We have interphase, which the cell is spending the majority of its time. It's when it's metabolically active and doing all the things the cell needs to do. And then when it's ready to divide, it's called mitosis. Okay, so that's just kind of showing you the cell cycle. We have interphase, which is huge, and then mitosis. And mitosis is further broken down into prophase, metaphase, anaphase, and telophase. Okay, so let's talk about interphase. Here, the cell is there for the majority of its life. Okay, it is kind of the period in between cell divisions. It's a period of growth. The cell is going to perform all of its metabolic activities and carry out all the preparations that are needed for cell division. Okay, so it is also broken down into G1, S, and G2. G0 is also a little subdivision, but it's not good if a cell goes into G sub 0. And I'll explain each of these as we go through the parts. Okay, so the main thing that happens in interphase is the genetic material doubles, so the chromatin replicates. We can see the nucleolus, or maybe there's many nucleoluses. We could still see that in the nucleus. We have a nuclear envelope is present, okay, and it's what we learned about the cell as far as the cell parts. So you can see the nuclear envelope, the chromatin, you have your nucleoluses, and so forth. And this is where our centrioles are. Okay, so let's talk about the different phases. Oops, sorry. So G stands for growth. So G1 is the first growth. So the cell that has just split needs to grow a little bit. S is synthesis. We're making more genetic material. We're making more chromatin. And G sub 2 is more growth. Now, I did say that if it went into G sub 0, that was very bad. So it's kind of like jail. It's an extended G1 phase. There are no cell divisions that can occur. The cells get stuck there. Now, not all cells can get stuck there, but the nervous system cells, adult nerve cells, do stick there. So when you do have a spinal cord injury, you typically don't heal yourself. Same thing with brain injuries. So if we have G sub 1, it can either get stuck into G sub 0, or it can go on to S, then it can go on to G sub 2, and then go on to mitosis. So G stands for growth. So growth, making more genetic materials, S. G2 is more growth, and mitosis is M. So it can get stuck in G sub 0. Again, this is just showing you a cell that has been in interphase. It's just doing all of its metabolic activities. It is preparing for cell division by doubling its genetic material. And these are the cells that are in interphase. All right, so now let's talk about mitosis. Mitosis is going to occur after G sub 2, and it is broken down further into prophase, metaphase, anaphase, and telophase. And telophase we're going to break down again into cytokinesis. Okay, so prophase. The centrioles are going to move to opposite ends of the nucleus. Your nuclear envelope is going to disappear. And your genetic material is going to go from chromatin to chromatids to chromosomes. So when we think of the main thing that happens in prophase, we're forming chromosomes. And you can see here the nuclear envelope still has to disappear, but we are forming chromosomes, and our centrioles are moving to opposite directions. Okay, you can see the centrioles here. They have moved to opposite directions. The nuclear envelope is gone, and we have made chromosomes. And this is what we can see when the cell. The nuclear envelope has to disappear so that the chromosomes have room to form. So this is kind of showing you a little early on. Okay, after prophase comes metaphase. Think of M for middle because the chromosomes need to move to the middle of the cell. And they do move by a molecule of kinesin. That kind of helps them get there. So that we do move the chromosomes to the middle of the cell. Okay, and you can see that they are lined up in the middle. Okay, so metaphase, the chromosomes move to the middle of the cell. And here's for the plant cell, they have moved to the middle. So after metaphase comes anaphase. So the chromosomes at this point are lined up at the cell. In anaphase, the chromosomes get pulled apart into chromatids, and those chromatids move in opposite directions of the cell. So the chromosomes are actually getting pulled apart. And you can kind of think of A as like an angry face, so that you can see the chromatids are making the teeth. And this is also going to cause the cell to change shape. The cell has to kind of stretch or elongate. And you can see here that the genetic material is now in chromatids, and they're getting pulled apart. Again, you can see that they're getting pulled apart. So this is early on, little division there, and then a little bit further. So anaphase, the chromosomes get pulled apart into chromatids. Early, and then a little later. Okay, so after anaphase comes telophase. Telophase is the reverse of prophase, okay? Because our chromatids are going to go back into chromatin. Our nuclear envelope is going to reappear, and in this case, we're going to have two nuclear envelopes because we have two different sides where the chromosomes have then pulled apart into. Okay? Now, for a animal cell, we get what this little cleavage furrow, this little indentation. Okay, so you can see that the cell is getting ready to split. For a plant, it's a little different. They don't get this cleavage furrow that we have here in the animal cells. So what we have in plants is called the cell plate because it does need to form the cell wall. So telophase is the reverse of prophase. We're going to form two nuclear envelopes, and the genetic material goes back into chromatin. Now, cytokinesis is the actual division of the cytoplasm. Okay, so it's the actual division. And you can see here by pictures that the cell has split. And those cells will go immediately into G sub 1 so they can grow and be metabolically active. Now, how do cells know if it's time to reproduce? Well, normal healthy cells contain a protein called the chalone. If they have them, they don't go through mitosis. If the chalone or that protein is missing, well, the cell knows that it does need to go through mitosis. And why would your body need to go through mitosis? Well, it would need to prepare for growth. It would need to maintain. Maybe you've gotten cut, your skin needs to heal, and so forth. If you have any questions, please see me tomorrow. Thanks.